So, hi everyone. Today we're going to talk about the commercial harvesting and the evolution of camouflage in alpine plants. So, I'm Selina.
I'm Jason.
Lindsey.
So, first we're going to talk about the background. So, first, we have to know what is the coloration. So, it's the key traits affecting survival and reproduction. So, it's common across the taxa. And then the second concept is about the plant camouflage. So it's always reduce the herbivory, but the role in avoiding human collectors is unknown. And also in animals, the human-driven selection, for example, the activities like hunting and fishing will impose strong selective pressure on morphology and coloration, so it's well documented. But for like plants, it has only limited evidence. So the commercial harvesting is unexplored. So that's why we have to focus on the F. delavii. Like this is the subject of the article. So it's the alpine perennial herb. And then for human, the bulb is for medicine. So it's high commercialized demand, and that's put a pressure. So make it very like intense and localized harvesting. And the leaf coloration also varies. It has green or like brown and gray color. So also like the nature herbivory on this species is rare. So this suggesting that there's non-nature selective force on it. And then for the knowledge gap, actually previous study has focused on like they have proved that the plant camouflage can actually reduce the herbivory, and also the background matching can vary among populations. But for like the color variation, whether it's because of adaptive or random phenotype variation is unknown. And also like human as like selective agent is also unknown. So that's why we have to make this study. And then the central hypothesis for this is commercial harvesting by human has driven the evolution of the cryptic leaf coloration in F. delavii populations. So for the study, the aim is to quantify color variation among populations and assess the degree of the background matching, and to test whether camouflage correlates with the harvest pressure, and also to evaluate the functional consequences of camouflage. Is a mess apart?
Alrighty, and to objectively quantify leaf and rock coloration, these researchers measured a reflecting spectra from 8 populations of Fritillaria delavii, as described in the published study. 12 to 27 leaf samples and 20 to 40 rock samples per population were collected. And using a spectrometer with a PTFE white standard for calibration, each sample was measured 3 times and averaged. Reflectance values between 390 and 740 nanometers were analyzed, corresponding to the visible spectrum for humans. Rock samples were collected within 5 centimeters of each focal plant to ensure that measurements reflected the true local background. Flower color was excluded because harvesting typically occurs after the flowering. And as we can see here, the results show significant color divergence among populations in a CIE lab color space. In this graph, can you go back? In this graph, each point represents an individual plant's color coordinates. The x-axis represents A values, which range from green to red. The y represents B values, ranging from blue to yellow. And camouflage populations cluster differently from green populations, showing a statistically significant divergence, confirming that color variation is not random but structured among populations. To statistically test the divergence, researchers converted reflectance data into CIE lab coordinates, which model human vision. They performed a MANOVO using LAB as dependent variables to test overall color differences among populations, and luminescence alone, represented by L, was tested using one-way ANOVA. The results showed significant divergence in both chromatic and luminance components with stronger divergence in camouflaged populations. This 3D plot visualizes how populations separate across LAP dimensions, and camouflage populations show tighter clustering near their background rock values, indicating adaptive background matching. The green populations show greater distance from local substrates. To directly quantify camouflage, researchers calculated the Euclidean color distance between each plant and its native rock background in CIE lab space. A lower color distance indicates stronger camouflage, and they also compared plant colors against non-native rock backgrounds to test whether matching was population specific. And here this graph shows us a significant negative correlation between color distance and collection intensity. Collection intensity was calculated as total harvested biomass divided by estimation population size. As harvest pressure increases, color distance decreases, meaning plants are better camouflaged in heavily harvested populations. This supports the hypothesis that human collection pressure is driving the evolution of cryptic coloration. And harvest pressure was estimated using two metrics. First, collection intensity based on harvest amount relative to plant abundance. Second, collection difficulty, measured as the time required to dig out a bulb with plants buried under tightly packed roots are harder to collect. And to test the functional impact of camouflage, researchers developed an online citizen science visual experiment where participants had to locate plants in photographs as quickly as possible. And here in figure C, it shows a positive correlation between color distance and collection difficulty. Plants that are easier to collect tend to be more cryptic, and this suggests that stronger harvest pressure selects for better camouflage. And here in figure D, in the online visual experiment, detection increased as camouflage improved, with targets with lower salience values meant better background matching, which took longer to find. Additionally, trichromatic viewers located plants faster than simulated dichromats, suggesting humans exert stronger visual selection pressure than most mammalian herbivores.
And then the article also compared similarities and differences of old findings versus new findings. And essentially what they found was that the camouflage of plants before high human activity used to be driven by the natural selection of herbivores. However, this study did not report any herbivory markings in our plant species of interest on the buds or the leaves. And humans were also in the past used to undergo selective hunting in ungulates, which is basically mammals with hooves, kind of like the deer we see on the right, which caused them to have a smaller weapon size. And weapons are basically their antlers or horns. And essentially, when we compare selective hunting to our plant species of interest, we see similar phenotypic changes. And then in conclusion, background matching in our species of interest is heavily correlated to commercial harvesting pressures. Humans have essentially driven the evolution of camouflage, causing the phenotype of plants to change for better concealment or hiding practices. And then why is this an issue? This is the big issue because now humans play a big role in driving the coloration changes and selective pressures on nature in the wild. Plant camouflage used to be a result of natural selection by herbivores when there was low human population, but now humans drive selective forces in areas where herbivores are rare, like in this study. And thank you for listening.
Thank you.
Thank you.